So good morning, students. Today, in the last class, we were talking about the chromosomal theory of inheritance. Okay, in that chromosomal theory of inheritance, we had studied how the other scientists, the work which was done by the Mendel was unknown till 1900. After that, some other currents, then some other scientists has came into existence, and they had worked more on that particular experiment. So according to that, they had introduced or they had discovered a theory that it is the chromosomal theory of inheritance. So in that, what they told me, so that is three scientists, D. Worries, that is Correns and von Desmarck. These are the three scientists. So what they told, they rediscovered the Mendel results on the inheritance of characters from one generation to another generation. So see here, in 1900, in 1900s, three scientists were there. Three scientists. On that day, I had mentioned the name also, that it is D. Worries, Correns, D. Worries, Correns, and one more, that is the von Desmarck. These were the three scientists who rediscovered the result on the inheritance of characters. Rediscovered the result on inheritance of characters. Okay. First, it was done by the Mendels. They rediscovered the Mendel's work on inheritance of what? Inheritance of characters. Okay. So this time, due to advancement in microscopy, as I told on that day also, due to advancement in microscopy, these three scientists, what they did means, they had searched once again on the what? On the Mendel's work. Okay, so that by taking place, scientists were able to carefully observe by using the microscopes and all. They carefully observe cell division. Okay, what they did, they carefully observed the cell division. Okay, so while they were carefully observing the cell division, at that time they had seen one structure inside that cell. Inside that cell, what they seen? They seen one of the structure, and that structure was the nucleus. Okay, that structure, what it is? What was it is? That it is the nucleus, which is the main component of the cell. In this nucleus, they observe. Okay, in this nucleus, what they observed? They observed some of the colored bodies. And what was their color? That colored bodies means it was the chromosomes. What it was? That it was the chromosomes. Chroma means color, somes means body. So in that he observed, they visualized by using some of the staining techniques or some of the stains. Okay, with the use of what? Microscopes, they carefully observed the cell division. In that they observed the nucleus, some structures they observed. With that, in that nucleus there was presence of what? Chromosomes. Okay, and in the year 1902, they observed the chromosome movement. What they observed? They observed the chromosome movements. the chromosomes are going to move. When? During the cell division. Which cell division means? That was the meiosis. On that day, I had explained you about the mitosis and the meiosis. What is the difference between the mitosis and meiosis? Okay, mitosis means that is a equational division. Meiosis means that it is a reductional division. Okay, so here the meiosis had been worked out. So while they were observing some of the chromosomes are going to move, in which division they observed? That it is the meiosis. Meiosis, which is the reduction division, as well as one cell, if this cell undergoes into the meiosis, this is going to produce how many daughter cells? That is the 4 cells. Okay, 4 cells. For example, how means if this is the diploid cell, if it is undergoing into the meiosis, there are 2 types of meiosis. That is the meiosis 1 and one more, that is the meiosis 2. Once they will, meiosis 1 is the reductional division by which the chromosome number is going to reduce. Okay, young. Then again it will undergo into the meiosis 2 division, that it is the equational division. So it will form the 4 number of, how many number? That it is 4 number of daughter cells. Okay, so they observe the movement of chromosomes during this meiosis. So one more scientist was there, 2 more scientists was there, Walter Sutton and Theodor Boveri. What was their contribution in his work? Let's see here. Here, 2 more scientists was there. Their name is Walter Sutton and Theodor Boveri. Already I had explained about this topic, but once again I am repeating this one. Okay, Walter Sutton and one more person, that it is the Theodor Boveri. These were the 2 persons noted that behavior of chromosomes was parallel to the behavior of genes. What they observed and what they noted that behavior of chromosomes and behavior of genes were parallel, means same. Okay, so noted that behavior of chromosomes was parallel to the behavior of genes. Okay, here parallel means The behavior of chromosomes and the behavior of genes were same. Who noted this one? This one was observed by the two scientists, that it is the Walter Sutter and the Theodore Boveri. Then next thing, what they told, and used chromosome movement. How they noted, how they observed means by what? By the movement of chromosomes. By the movement of chromosomes, they observed that the behavior of chromosome was similar to the behavior of what? Genes. Okay. To explain Mendel's law, so here during mitosis, what they explained? They explained both mitosis and meiosis. So during mitosis, what they observed or what they told on Mendel's work means during mitosis. As you know, mitosis is also known as the equational division. Equational division. Okay, during mitosis and during meiosis. One more division, that is meiosis. Just now I told you, which is also known as the reductional division. Okay, when they observe the movement of chromosomes during mitosis as well as during the meiosis. Okay, so the important things here to remember in the mitosis and meiosis, that chromosome as well as the genes occurs in the pairs. How they are present? They are present in the pairs. Here I will show you. See, in first year class you had studied about the cell cycle. In this cell cycle, there is a presence of phases. This is the S phase, and after S phase, in this S phase, that is the synthesis phase, it has three phases. Sorry, this is the I phase. Interphase. This interphase has three phases: G1 phase, S phase, and G2 phase. Okay. The next, the cell cycle will have the M phase, that is the mitotic phase. In this, first is prophase, metaphase, anaphase, and one more, that is the telophase. Already you had studied. Okay, so this is the M phase, and this is the interphase. G1, G2 means that these two are the gap phase, where the DNA or the cell is going to prepare for the cell division. Okay, gap 1 phase and gap 2 phase. S phase means that it is the synthesis phase. Okay, so what they are telling, whenever the division is going to take place, the genes and chromosomes will be present in the pairs. Okay, so here you see, on that day, I had given two pairs of chromosomes. Here you see, during G1 phase, how it will be. This is one pair of chromosome. Okay, this is another pair of chromosome. This is one pair and this is the another pair. Like this it will be. Why I am shading here means to differentiate them. Okay. In NCERT book, they had given in colors. Okay. In spite that one, I am shading here. This is one pair and this is the another pair. The chromosomes will be like this during the G1 phase. Okay. Then when the G2 phase will come, the chromosomes will occur in the pairs like this. because the DNA duplication is going to take place, the DNA replicate and the DNA is going to synthesize. See here, for example, when the DNA will be like this in the G1 phase, in the G2 phase, during S phase, this phase, the DNA is going to duplicate. Means here it is going to duplicate, means they will form a 2 number of DNA. Okay, so the chromosomes are also going to occur in the pairs. Okay, so here during G2 phase, the chromosome will occur in pairs. Like this. Okay, because in between the G1, this is about the G2 phase. In between the G1 and G2 phase, there is a S phase, during which the DNA will make its own copies. Okay, when DNA is making its own copy, means the chromosome also doubles. Yes, sir. The chromosome also doubles. Here it is singly present, there it is present in pairs. Here also you see. In this manner, the chromosome will appear when the G2 phase is going to occur. Okay, then comes bivalent are going to form. Bivalent means where the two sister chromatids are going to they are going to come closer to each other. For what? Crossing over. That we can call it as the bivalent. Here like this. You can see how the chromosomes are coming closer to each other. Okay, you see how the normal. Then this after that, during anaphase, this chromosome will move towards the opposite poles, as the centrome here is going to divide like this. Okay, they will move towards the opposite poles. So this is the anaphase. In this way, they are going to separate from each other. When they are going to separate, they will form like this structure. This is the anaphase of meiosis 2. As I told you, meiosis is divided into two types. Meiosis 1, which is the equational division. Meiosis 2, that is the reductional division. Here, pairs you can see. Here, individual chromosome you can see. Like this. Okay, means here you can see. Here they are present in pairs. Here they are moving towards the opposite poles. Okay, so there you can see a single chromosomes when during the meiosis 2. Here also, totally they will get separated from each other in this manner. Okay, separated from each other from the centromere point. Then next, when the germ cells are going to form, gametes are going to form, you can see this is the gametes, where the 4 chromosomes are going to be separated. This is one germ cell, this is the another germ cell. This is the third one. And this is the fourth one. In this manner, 4 germ cells are going to produce, in which there is an equal distribution of what? Of chromosomes. This is the behavior of what? Chromosomes. Okay. On these chromosomes, there will be presence of what? Genes. Okay. If chromosomes are just dividing in that 4 germ cells, then genes are also going to divide there. They are also going to occur when chromosomes. See here the chromosomes are in pairs. Then the genes will be in the pairs. Okay. When see chromosome on the chromosomes, there will be presence of genes. Okay. If this chromosome is going to separate it, here we see, then genes are also going to separate it. See, for example, this is one gene, this is the another gene. When this chromosome is going to separate it during anaphase, the genes are also going to separate it like this. There you can see. Okay, that only they are going to show here. The next topic, we have to see the comparison between the behavior of chromosomes and genes. What are the comparisons? So let's see. Next, comparison between the behavior of chromosomes and genes. Between chromosomes and genes. So here one table is there, that is behavior of chromosomes. Here is behavior of genes. So let's see, or let's compare the behavior of chromosomes and the behavior of what? Genes. Okay. First of all, either it may be chromosome or either it may be genes. They both will occur in pairs. Okay. First point, occurs in pairs. Okay. Here also, if chromosomes are occurring in pairs, so see here, if chromosomes are present like this, means in pairs they are present means the genes will also present on them. They will also occur in pairs. See here, this is the chromosome. On this there is a presence of genes. If they are only present in a pairs means, then the genes will also occur in pairs. See, here also occurs in pairs. If chromosomes are in pairs, then genes are also in pairs. Then, first, second point. Segregate at the time of gamete formation. Segregate at the time of gamete formation. What it is going to segregate? That it is the chromosome. Such that only one of each pair is such that only one of each pair is transmitted to a gamete. This is about the chromosome. The next we will see about the genes. Segregate at time of, second point. Sorry, this is the first. This is the second. Segregate during gamete formation. Gamete formation, and only one of each pair, and only one of each pair is transmitted to a gamete. Okay, so see here, this is the prophase in which the centromere will not there, only the chromatin fibers will be there, like this. This is the prophase, and this is one of the diploid cell, which is undergoing into the meiosis. Okay, this is the, here I will write meiosis 1. Okay, so during meiosis 1, this is the prophase or this is the diploid cell in which the chromosome will be fat-like structures. Then this is the metaphase. After prophase, there will be presence of metaphase, means the chromosome will lie at the equatorial plane. That too, they are in pairs. Okay. The next during anaphase, they will segregate from each other in this manner, or they will move towards the opposite poles. Okay, like this. Then during telophase, two cells are going to form. One pair will be present in one cell, and the another pair will be present in another cell. This is the telophase. Meiosis 1 will, one cell will give rise to the two new cells. Okay, as you know, meiosis 1 is the reductional division. Here, if this cell is diploid, here they will form haploid, haploid. These cells will be the haploid one. Then again, these cells will undergo into the meiosis 2 division. Same thing, the prophase, anaphase, metaphase, sorry, prophase, metaphase, anaphase, and telophase. After that, these two cells will give rise to the 4 cells, and that 4 cells will have a particular, means the chromosomes are going to segregate, and one chromosome is going to transmitted or distributed to each of these cells, and this all 4 cells will be the haploid one. Okay, so in this manner, what they are doing, they will segregate, in separate. The chromosomes are going to separate. Here you can see how the chromosomes are going to separate. Here they are present in pairs. In metaphase, you clearly can see they are present in pairs. But during telophase, they are going to separate. But during meiosis 2, totally they are separated. At the time of gamete formation, especially at the time of gamete formation, they are going to separate it such that only one of each pair is transmitted to the gamete. Okay, either it may be chromosome or either it may be genes. Both are going to be separated during gamete formation, and that only one pair is going to transmitted to the next gamete or the germ cell. Okay, then third point, you see. Independent pairs segregate independently. Independent pairs segregate independently. Okay. independently of each other. This is about what? About the chromosome. Then here, one pair segregate independently. One pair segregate independently of another pair. Means here in the Mendel's second law, I had told you that law is known as the law of independent assortment. Mendel's second law, that is law of independent assortment, where the pairs of chromosomes, they are going to separate independently of each other. Okay, here independent pairs segregate, means they are going to separate from each other independently according to which law? That is the independent assortment. Okay, and as well as the genes, which where one pair segregate independently of another pair. Okay, so this is about the comparison between the behavior of chromosome and behavior of genes. Means the behavior of chromosome and the behavior of genes, both are similar. All the points are similar in the chromosome as well as in the genes. Okay, the next here, see, Sutton and Bovary had given this comparison between the what? That it is in behavior of chromosome and behavior of genes. They argued, means they had debate on what? On the pairing and separation of pair of chromosomes. So that leads to the segregation of pair of factors, that it is the genes. The genes are also going to separate it and the chromosomes are also going to be separated. Then Sutton united the knowledge of chromosomal segregation with Mendel's work or Mendel's principle, and they had given the name to this theory as the chromosomal theory of inheritance. Okay, so this is about what? About the chromosomal theory of inheritance. Then next, what is the next topic here? Means that is the linkage and recombination. In that, one scientist is there whose name is T.H. Morgan, who had experimented on the fruit flies. Okay, that fruit flies, which is also known as the Drosophila melanogaster. Why he had selected fruit flies only? Then what was his experiment and how he had explained the linkage and recombination? That we will see in the next class. Okay, thank you.